This recording is going to cover some major events that take place in your distal tubule, but also the collecting system, the cortical collecting tubule as well as the medullary duct. So I'm actually going to break up the distal tubule to three major components because there's just a little subtle differences between them. Now, the early distal tubule as well as the distal convoluted tubule will have similar things that take place as we have in the thick ascending limb of the loop of Henle. They're impermeable to water. You have reabsorption of ions such as sodium and chloride and even calcium. But a subtle difference with regards to calcium is most of your calcium is actually reabsorbed in the proximal tubule over here, about 65 to 70%. 25% is reabsorbed in the thick ascending limb, but both of those are reabsorbed through the paracellular route, so between the cells. What's left, which isn't a huge amount, maybe up to 10%, is actually regulated by the parathyroid hormone. And the calcium is reabsorbed through the transcellular route, so across the cell. And parathyroid hormone regulates this. So if parathyroid hormone's around, you'll have a lot more of these calcium channels as well as this calcium pump over here, which allows us to reabsorb more calcium. So it's going to be where it's going to be regulated. So that's a little difference between the early distal tubule, distal convoluted, compared to earlier on in regards to calcium. Now, the one thing, though, about the early distal tubule that's different from the other parts of the distal tubule is this is the location, and this is not right, I'm going to put early distal tubule. This is where you have those macula densa cells. So the macula densa cells are a component of your juxtaglomerular apparatus. So the juxtaglomerular cells are those smooth muscle cells associated with the afferent arteriole. Those two together are very important in the regulation of your GFR. So remember, the macula densa cells actually have sensors that detect the concentration of sodium ions in the tubular fluid. And based on what happens to it, it can tell if your filtration rate has gone up or gone down, and it will communicate with the juxtaglomerular cells. And the amount of renin that's released from that would be very important ultimately in the regulation of your GFR. Now, the late distal tubule, and I'm going to include the cortical collecting tubule because they have similar things that take place here, this is different. The late distal tubule is different from the earlier parts of the distal tubule in that one of the things that we're going to mention is can be permeable to water dependent upon ADH levels. Plus, these are going to be important. You have these principal cells that will be important in regulation of sodium and potassium balance because they're regulated by aldosterone. So they have receptors for the hormone aldosterone. And so aldosterone, being a steroid hormone that it increases the amount of the sodium-potassium pump here that we have in the basolateral membrane, and then it increases the insertion of sodium channels and potassium channels over here in the luminal surface. As an end result of aldosterone is you'll have an increase in sodium reabsorption and an increase in potassium secretion. Another type of cell that we find in the late distal tubule as well as the cortical collecting tubule are intercalated cells. Now, this is only showing you one. There's more than one type of intercalated cell. So depending on the situation, you can regulate the amount of hydrogen ions that are going to be secreted and the amount of bicarbonate that's going to be reabsorbed. And this situation you see here is we have this hydrogen ATPase, which is going to be involved in secretion of hydrogen ions. And that would be something we'd definitely want if you're acidotic. We want to get rid of excess hydrogen ions. At the same time, I want to be able to reabsorb some bicarbonate because it's actually important as a buffer. But depending on the situation, the type of cell we'll talk about at a later date, is we can have increased hydrogen ion secretion or decreased, or increased hydrogen ion absorption or reabsorption or excretion. So it depends on the situation. But these two types of cells, the principal cells and the intercalated cells, are found both in the late distal tubule as well as the cortical collecting tubule. Now, I had mentioned previously is the permeability to water is variable in this part, the nephron tubule, is in response to ADH. So ADH, the receptors found in these parts of the nephron tubule, as well as, I'm going to include, as well as the medullary ducts, in response to it, through its second messenger system, what it does is it increases the amount of these aquaporin 2 channels that we have over here on the luminal membrane. That's going to allow water to move into the cell, and we have these aquaporin 3 channels over here on the basolateral membrane. And the end result is, if we have more of these aquaporin channels, we'll be able to reabsorb more water. And so this part of the nephron tube is very important when we talk about regulation of urine volume. It's the sensitivity to ADH will determine if we're going to reabsorb a lot of water or not so much water. Now, the medullary duct is part of that collecting system, like the cortical collecting tubule. So I mentioned previously, the cortical collecting tubule has the principal cells as well as the intercalated cells, like that late distal tubule. So again, principal cells are involved in sodium and potassium regulation, because they're sensitive to aldosterone. Intercalator cells are very important in acid-base balance. And these cells, again, are sensitive to ADH. So the amount of ADH will determine how permeable it is to water. Well, the medullary duct is also sensitive to ADH. And another thing, though, what's going to be important is the ADH, besides increasing the permeability to water, it increases the permeability to urea. And you're like, why would we want that? Urea is a waste product we want to get rid of. Thing is, the urea is also a major component of the interstitium, in which we have this osmolarity, steadily increased osmolarity as we go from the cortex down to the deep medulla, is we have the osmolarity steadily increasing. And what creates that osmolarity that we see is sodium and chloride, but also urea. So in instances where we want to concentrate your urine, we want the osmolarity pretty high down here, and urea actually contributes about 50% of that. And so, but don't worry, eventually we'll get rid of it. It's just there. We're going to utilize it to our advantage to be able to concentrate our urine. So kind of like in a nutshell, that medullary duct is still reabsorbing ions, and it can reabsorb that urea. Urea always moves passively. You have reabsorption also of bicarbonate here. You can have secretion of hydrogen ions, which will be important again with acid-base balance. And again, it's permeable to water with more permeable to water with the presence of ADH. So depending on the situation, is if that medullary duct, say for example, was impermeable to water, you don't have a lot of ADH, you still have reabsorption of various ions, but water can't leave. And that's why you end up with a very dilute urine, okay? But if ADH was present, we'll be able to, we'll erase this. This is permeable to water, and so as you're reabsorbing those ions, you're also reabsorbing water. But I had mentioned is the osmolarity steadily increases in the interstitium in the medulla, the renal medulla, and so it's going to draw more and more fluid out, and then my urine volume would be a lot smaller or more concentrated. We're going to look at that in a subsequent recording. But one last thing I do want to mention is we do have hormonal control of some things that in the nephron tubule. So I had mentioned about aldosterone. That regulates, and it's not just cortical collecting tube, but that's also the late distal tubule. That is important in the regulation of potassium and sodium. If that part of the nephron tubule is permeable to water, water will also be reabsorbed. So if ADH is around, you'll also reabsorb water. But that just comes along passively. Angiotensin II affects sodium chloride absorption. And the big one I want you to remember is in the proximal tubule. It does also will affect hydrogen ion secretion, but you don't have to worry about that. ADH is a big one. That affects the water reabsorption in the distal, late distal tubule in that collecting system. Don't worry about this one, but parathyroid hormone. That regulates calcium reabsorption. Now here, it's not that, it's not that. It's the late, not late, it's just distal tubule. We'll just say distal tubule. So parathyroid hormone regulates the reabsorption of calcium by affecting calcium transport in those parts of the nephron tubule. So cross those out. And it will also affect phosphate reabsorption, so it decreases it. We have talked about this at a previous lectures, but of these, the ones that I want you to remember is definitely aldosterone, ADH, and parathyroid hormone. And the only thing about angiotensin II, it does increase absorption of sodium in the proximal tubule.